Hello to our friends joining us on our recording. Today, we are covering lab lesson number 2. There are 3 topics we're going to cover today. We'll take a little bit of time to talk about regional terms. Then we'll spend a lot more time talking about how to use microscopes. And then finally, we're going to cover cytology, which is the study of cells. So as we mention our first topic here, regional terms. At the very beginning of your lab packet, you have a list, one page that includes tons and tons of text, right? That shows you descriptions of body regions for you to match up with the words. You also, though, have these pictures here that show the front side of the body and the back side of the body. Here's what Dr. Allis thinks you should do when you're studying these regional terms. I don't think you should spend almost any time at all on matching up words with their descriptions of where we find things. What I want you to make sure you spend your study time doing this week is being able to find these regions on pictures or on yourself. So when we talk about regional terms today in our class, we are only going to talk about what we see here on our pictures. So with that in mind, let's go through and talk about some of the regional terms that we need to know. When you're trying to find body regions, some of the regional terms that you see on the list are going to look a whole lot like bone names. So, for example, let's look at this region right here in the very middle of your chest. We needed to know the name of the bone that lives right here in the very middle of the chest. Does anyone remember the name of this bone that lives in the very middle of the chest? Exactly. Yep. Several of us chiming in. This bone here in the middle of the chest is called the sternum, the central breastbone, if you will. The sternum is found inside the part of the body that's known as the sternal region. If I know where the sternum is, I can find the sternal region. Hey, when I'm talking about the region called the femoral region, what bone do you think I'd find the femoral region next to? What does that sound kind of like? Yeah, exactly. The femoral region sounds like the femur because that's where it is. It's right on top of the femur. Hey, some of those regional terms are going to make us remind ourselves where we find some of the trickier bones in the body. So a couple of those regions I want to do with you have to do with the radius and the ulna in the palm of the arm, and the tibia and the fibula that we find down here in the leg. So this gives us a chance to review our bones a little bit. Up here in the forearm, you have a radial region and an ulnar region. The radial region is on top of the radius. The ulnar region is on top of the ulna. To help us figure out which one is medial, which one is lateral, let's look at our picture here and remind ourselves. When I use this word medial, remember that medial means it's toward the middle of the body. So here's my medial region right here. Incidentally, my medial region is also on the same side as my pinky. Was it the radius or the ulna that's found on the same side as the pinky? The radius or the ulna? Do we remember? Exactly. Yep. So the ulna is the bone that we find on the pinky side of the arm. Remember, the way that Dr. Allis remembers that is we have the word up. Ulna, pinky, up. So the ulnar region would be the medial side of the forearm, the side that's closer to the middle of the body, or the side that's the same as the pinky. So the radial region is going to be the lateral side of the forearm because it's on top of where the radius would be. Hey, when we go down here into the legs, the 2 bones that we have in the legs are the tibia and the fibula. Remember that the tibia is the T-bone and the fibula is the really skinny bone. Is it the tibia or the fibula that's on the middle side, the medial side of the leg? Do we remember? Tibia or fibula? over here. Exactly. The big T-bone is on the medial side of your leg. The little fibula, like a little fib, found on the outside of your leg. So the lateral side of the leg, that's the fibular region. The medial side of the leg, that's the tibial region. So here's what we've talked about so far. We've talked about using the names of bones to help us figure out where some of our regions are in the body. If a name looks like a bone, it's found by that bone. The next clue that we can get from our body regions is thinking back to those cavities that we talked about last week. So we have a region in the body that is encasing the whole chest cavity. Do we remember from last week? What was the name of the big cavity that made up the entire chest? What cavity is up here in the chest? Exactly. Yep. This big area up here was the thoracic cavity. It's also the thoracic region. So just like this up here was our abdominal cavity, here's that abdominal region. Or the pelvic cavity down here, the pelvic region down here where we found that pelvic cavity. So look for something that looks like a bone name. That'll help you find it. Or think about those body cavities that we talked about. There's a couple others that I'll point out to you that maybe aren't quite as intuitive because they don't match up with things quite as well. So a few that we're just going to have to memorize. One of them would be this region right here, the armpit region. This is called the axillary region. So we could take an axillary temperature by putting a thermometer in your armpit, leave it there for a while, see what it says. So the axillary region, the armpit region. I also want to mention this region over here by your hips. We learned the names of each of the hip bones, but sometimes we refer to them together in a group. I'm going to type in the chat what we'd call them as a group. As a group, all of the hip bones together are called os coxae. So when we talk about this region here, the hip region on either side, we would call that the coxal region, the hip region based on those os coxae. So when you're studying these things, any tips or tricks that you can use for yourself, any ways you can connect these regions to the names of bones that you learned or to the names of cavities that you learned, that's going to help you out. Make sure, especially on the ones though that you've never heard these words before or seen these words before, those are the ones to make sure that we really memorize. We practice those ones a lot. And again, don't spend a lot of time working on matching up these descriptions. I don't want you to make flashcards with these regional terms with a definition of what they mean. Unless there's a few that when you're working on labeling them on the body, they give you a lot of trouble. If you really have trouble with the hips being that coxal region, make yourself a flashcard of that, but don't make a flashcard of everything else. You'll waste a lot of time making flashcards over stuff that you already know. And as you've become acutely aware already here in week 2 in anatomy, we don't have time or mental space to memorize everything. Some stuff we're going to have to get into our long-term memory. Some stuff we're going to have to predict or use our bodies or use pictures to help us out with. So don't spend too much time on these descriptions. Do spend a lot of time finding those regions on your body and on pictures. I want to spend most of our lab time today talking about the 2 other topics in our lab. So the 2 other topics are microscopy, which is using a microscope to help us see things that are small, as well as cytology, which is the study of one of those small things. In particular, we're talking about cells. So remember how in lecture on Monday, we talked a little bit about organelles and what they do and how we use them in a cell. So we're going to build on that today and start to identify those organelles and remind ourselves about what they do. When we talk about microscopy, We need to go through and start to learn about the different parts of a microscope. Because remember, we're going to start using these microscopes next week. It'll help us to know what our different parts do. So let's go through describing these parts of the microscope in the way that you would encounter them in a typical lab class. At the beginning of a class where we are using the microscopes, we have to go retrieve them from a cabinet and bring them to our lab bench. To help you carry the microscope, there are two parts that assist with this. So the first part is this right here, the part that's going to sit on your lab bench, labeled with my arrow right here. This is the part of the microscope called the base. So the base of your microscope is the very bottom of it, what rests on your lab bench. The other part we'll use to transport it is this part that sticks up right here, labeled with an arrow right here. This is the arm of the microscope. So the base and the arm, you'll put one hand under the base, one hand on the arm. Those two things are where you'll be gripping to help you transport the microscope. When you get the microscope to your lab bench, you're going to look into that microscope using this part that's sticking up here. This is called the oculars. So the oculars are the part of the microscope that you look through to identify specimens. I'll mention that the oculars by themselves can make things 10 times bigger. So if we took the ocular off of the microscope and we put it by our eyes and peeked through it, anything we looked at would be 10 times bigger than it is in real life. The oculars, what we look through, make things 10 times bigger. But 10 times bigger by itself is not usually enough to help us see what we need to see. And that's why we need to use these things that we see on our picture right here. These things are called the objective lenses, and they each have their own name, and they each magnify things in slightly different levels. So let's start with our smallest objective. We see it right here with a red stripe on it. The smallest objective is called the scanning objective. So the scanning objective, my one with the red stripe, this magnifies things 4 times. So again, by itself, it doesn't make them much bigger. We'll talk about, though, every time you look at something in a microscope, it's actually being magnified twice, both by this ocular up here at the top and by whatever objective you're using. So my scanning objective makes things 4 times bigger. Not super impressive on its own. Next door to the scanning objective, we have what's called the low power objective. The low power objective by itself magnifies things 10 times. So another 10 times magnifier right here, that low power objective. The high power objective is my last one that's seen here with the blue stripe. The high power objective by itself magnifies things 40 times. So this is the big guns over here. They've got lots of magnification power, especially when we pair that with our ocular that helps us to magnify things by itself. When you're looking at slides in lab, you will almost always use more than one of these objectives to view that slide. We always start with the scanning objective in place. We always start with red, and then we'll at least go up to yellow, if not also all the way up to blue. So to help us be able to trade objectives, we have a part labeled with the arrow right here. This part is called the revolving nose piece. Like its name suggests, the revolving nose piece turns in a circle. It revolves. So you can decide which of these objectives is pointing down based on what you're trying to visualize. So again, we always start with the scanning objective in place on that revolving nose piece pointing down, and we'll go up from there as we need to see things bigger. When we are looking at a slide, we're always going to place that slide on this flat black part that we see right here. So this flat black part that we see right here, this is called the mechanical stage. And we can see the mechanical stage over here as well from the side. This flat black part, the mechanical stage. The mechanical stage is what your slide sits on top of. The mechanical stage can move up and down. We'll talk about how we make that happen. But the big idea is that stage is what holds your slide, what keeps it in place. Sometimes when we put a slide on the stage, we need to move it left or right or forward or backwards. To enable us to be able to do that, we need to put the slide into this metal thing that you see right here. This metal thing that we see right here is called the stage clip. And I know it's a little bit funky and harder to see, but this arrow over here that's pointing, this is also actually showing us the stage clip. So the stage clip is right on top of the stage. Here's the button we'd press to open or close it. And that will allow us to put a microscope slide here in the middle of that clip. Once your slide is clipped in place, we can use the stage adjustment knob that's hanging down from the stage right here to move that slide forward and backwards, left and right. So the stage adjustment knob right here. So I've got my slide clipped in to my stage clip. I've moved it around with that stage adjustment knob so that my tissue is in the light. Now I need to make it, what is a blurry image, become more clear. Now I'm going to go over here on the arm of my microscope and I'm going to start focusing it. So first, I'm going to use my coarse adjustment knob. That's the big one that's really close to the arm, the coarse adjustment knob. I'm going to use the coarse adjustment knob to get it almost all the way in focus. Then I'm going to switch to my fine adjustment knob to get it 100% clear. The coarse adjustment knob moves the stage a lot. It goes up and down a lot. The fine adjustment knob barely moves the stage, but it moves it just enough to help you bring things into focus. We don't make you label the two adjustment knobs on this picture, but I'll mention this one right here that's closer to the arm. That's the coarse adjustment knob. And this one right here that's sticking out from the arm, that's the fine adjustment knob. So the coarse and the fine adjustment knob helps us to get things into focus. Here's something that I need everybody to write down somewhere on your paper. So find yourself a space where you can write a statement. I have an underline highlight star idea. I have something that's really important for all of us to write. Here's what all of us need to write down. You can only use the coarse adjustment knob when the scanning objective is in place. You can only use the coarse adjustment knob when the scanning objective is in place. What do I mean by that? I mean that when my red objective is pointing down, I'm allowed to move my stage up and down all over the place to get it in focus. But once I rotate my revolving nose piece and my low power objective is pointing down, once I rotate that over, I am not allowed to touch that coarse adjustment knob anymore. Now I can only use the fine adjustment knob. And especially when that high power objective is in place, I can only use the fine adjustment knob to adjust things. Let me introduce a concept for you that explains why we can only do those things. So I'm going to draw for you here a little bracket. This bracket is representing the amount of space between the end of my objective and where my slide would be. So the space between the end of my objective and where the slide would be, this is something called working distance. I just put it in the chat for you. Working distance. That's the amount of space between the objective and the stage. When I have the scanning objective pointing down, my working distance is really big. The space between that objective and my slide, lots of distance. But once I go from the scanning objective to the low power objective, the amount of space between them gets smaller. And when I put in that high power objective that's really big, it's almost scary how small my working distance is. The reason we cannot use this coarse adjustment knob when the low power or high power objectives are in place, when yellow or blue is pointing down, is because it'd be too easy for us to accidentally move the stage so high that we break our slide. So we can only use the coarse adjustment knob when we have the red pointed down. There was a good question in the chat when we're talking about the fine adjustment knob. Yeah, I can use that anytime. So I can use that starting with when the scanning objective is in place. I get it mostly focused with the coarse adjustment knob. And then I fine tune it, get it totally focused with that fine adjustment knob. Now I'm ready to go up to yellow. And once I'm on yellow, I can only use the fine adjustment knob, get it in focus, go over to blue. Fine adjustment knob, get it down. So the fine adjustment knob is fair game with any of the objectives in place, but the coarse adjustment knob can only be used when we have the scanning objective in place. One other idea I'll mention with that, the other reason that you don't want to be using the coarse adjustment knob when we go above the scanning objective is because our microscopes are parfocal microscopes. Parfocal means if you do a good job getting it focused with the scanning objective in place with red, then when you switch to yellow, to low power, it's mostly already going to be in focus. So I should just have to do a little bit of tweaking to get it all the way in focus. And if I get it all the way in focused on yellow, then I should have to do just a little bit of tweaking to get it in focus on blue. So parfocal microscopes mean do it right at the beginning on red. That'll help you with yellow. And if you get it right on yellow, that'll help you with blue. I will tell you in lab next week and in the many weeks to come where we spend a lot of time on microscopes, I will tell you if you can't get it to focus on blue, go back to yellow. We got to make sure it's focused on yellow. If you can't get it to focus on yellow, go all the way back to red. We got to start over, get it in focus on red. Because if we don't focus it on the scanning objective lens, nobody else is going to work. It's a parfocal microscope, so it's kind of like all or nothing. Either we're set at the beginning and we're good all the way up, or we're completely messed up from the beginning to the end. Parfocal microscopes. Let's talk about a few other microscope parts briefly. Here underneath the mechanical stage, you can see the light source for your microscope. For you to be able to see anything, we need to illuminate it. So here's that light source. Down below that light source, you can see this circle right here, this knob. This is the light adjustment knob. The light adjustment knob allows you to make the light brighter or darker. So it's kind of a crude way to change the amount of light that is available to potentially light up your slide. The better way that we change the amount of light that is going onto our slide is using this thing right here. So this whole thing that we see hanging out underneath the stage is something called the iris diaphragm. The iris diaphragm works just like the iris in your eye. So that's the part, the colored part that gets smaller or bigger, depending on how much light is there. So we can use this iris diaphragm lever to open or close the iris on the microscope. That will help us to determine how much or how little light goes to the microscope. I'll mention there's one other part that you don't have to know where it's found, but you do need to know what it does. And it's a part that lives up here, right, kind of above the iris diaphragm. It's a part called the condenser. The condenser. And the condenser is what takes whatever light gets through the iris diaphragm and condenses it or focuses it onto your slide. So again, we're not going to ask you to show us where the condenser is. For your reference, it's up here above that iris diaphragm. But the job of the condenser is really important, focusing that light onto your slide. I have talked for entirely too long. I need to open the floor for questions or for emojis. What questions do we have? What emotions are we feeling? Let's take a moment to pause. Getting a couple of smiles. Saw some thumbs up. Good. Yeah, Jamel's about done. I feel you, Jamel. Hey, the good thing about online classes, right, is Jamel could actually be laying in bed and we wouldn't know, right? So that is the benefit of classes from home, at least. We've got a question. What is the name of the thing above the iris diaphragm? Let me put that to the class. What did we say the name of this thing was? Do we remember? Anybody catch it? I know I said it fast. Yeah, so this is the iris diaphragm lever. So the whole thing here is the iris diaphragm. This is a little lever that will move back and forth to open or close that iris. Yeah. I'm glad to hear it. Jamel has made sure we all know not in bed. That it's better that way. You can focus better when you're not laying down, right? Although it is more fun to lay down for sure. Any other questions before we move on? I think we seem to hit it pretty good. All right. Well, let's talk a little bit more about microscopes. And then I'm going to actually take you to a virtual microscope that I'm going to show you how it works together. So uh, I'm not going to take time to match these. We talked through these functions. You got the guided lesson. What I want to talk to you about or remind you of, shall we say, since we talked about this before, is that we have those two adjustment knobs, the coarse adjustment knob and the fine adjustment knob. Depending on which color objective is pointing down, that'll help us to know which of the knobs we can use. So first question, I'm talking to you about the scanning lens. Can you remind me in the chat, what color is the scanning lens? What color was the scanning lens? Yeah, so the scanning lens is the red one. It's the smallest one. It only magnifies things 4 times. When I have that scanning lens in place, when I have the red one down, can I use coarse, fine, or both adjustment knobs? Coarse, fine, or both when that red one's pointing down? Exactly. When the scanning lens is in place, anything is fair game. You always want to start with the coarse adjustment knob because that'll get you mostly in focus faster. Then you'll move to the fine adjustment knob to get it all the way in focus. And remember, once we put down the yellow, which is low power, once we put down the blue, which is high power, we can only use the fine adjustment knob. So we'll practice this in lab next week. But again, the only time I can use that coarse adjustment knob is when I have that scanning lens in place. Our microscopes are what we call compound microscopes. When we talk about a compound microscope, that means that when we magnify things, we always magnify them twice. So the first thing that's making what you see bigger are those oculars. So oculars, remember, are the things that you look through. By themselves, the oculars make things 10 times bigger. But then the other thing that's magnifying things, like we've talked about, are the objectives. And remember that we actually have 3 different objectives that are different colors and have different magnifications. So everything that you're looking at on your microscope slide, when you look through those oculars, is magnified by both of these things, which means that we have a total magnification that's bigger than either of the magnifications before it. I won't make you do much math in our class together this semester, but I am going to make you do the total magnification math. I promise, though, it's not too scary. We can all do this. I believe in us. When we're trying to figure out how much bigger what we're looking at compares to the size of what it is before we put it on our microscope, we have to multiply how much bigger the ocular makes something compared to how much bigger the objective makes it. The good news is my oculars always make things 10 times bigger every single time. So the first number is always the same. We're always multiplying by 10. My objective magnification is going to depend on which one is pointing down. So let's see if we remember. We had the 3 colors on our objectives: red, yellow, blue. When that red objective is pointing down, do you remember how much bigger the red one made things by itself? My little red one. I feel like doing like the Southern Texas thing, like, oh, bless its heart, the little red one. Yeah, everybody's right. Little red makes things 4 times bigger. Not too impressive, right? But it's something. When that yellow one, the low power, is in place, that's my middle one, right, that I see on the list. That's 10 times bigger. Things are 10 times bigger. And that blue one, that by itself makes things 40 times bigger. 40 times bigger. So now I got to break out the math, right? I got to do my mathematical literacy here. So I've got my red objective in place. My scanning objective is down. What I'm looking at through my oculars is 10 times 4 times bigger. So I'm looking at something that is 40 times bigger than normal. Most of the things we look at, making them 40 times bigger is not helpful enough. We got to go bigger. So I slide my low power objective into place. That's my yellow objective. Now I'm making things 10 times bigger with the ocular times 10 times with the yellow objective. Now I'm making things 100 times bigger. 100 times bigger. This will be big enough for us to look at things like bone tissue or nervous tissue. But when we're talking about things like epithelial tissue, which is most of the tissue in your body, we're going to have to use that blue objective. That blue objective by itself made things 40 times bigger. So 40 times from my objective times 10 for my ocular means what I'm looking at is 400 times bigger than what I'd see in real life. We really get our power the higher up we go in those objectives. That's where the magnification power comes from. So there's 2 ways for you to approach this total magnification thing. Way number 1, if the math isn't too scary for you, you can just do this calculation when you're doing the homework or the quiz or the exam. But number 2, if the math is scary for you, you can just memorize these numbers. You can just memorize that the total magnification with the scanning objective, 40. The total magnification with the low power, 100. 400 for that high power. So keep that in mind, 2 ways to study. Any questions about total magnification before I move on? This is when I put in the crickets, right? Crickets are probably a good thing. Probably. Otherwise, it means that everyone left and Dr. Ollis is talking to the void. And that would be sad. All right. There comes the cricket. I like it. All right. So we've talked about total magnification. Let's talk about the difference between magnification and resolution. Everything that we look at on the microscope, we're always going to make things magnified. Magnification just means you're making it look bigger. The microscope always makes things look bigger. But we don't just want things to look bigger, we also want them to be clear. So resolution is making things clear when we're looking at them. So let's say that I've started with this letter E that I can see right here. Both of my pictures show the letter E that's magnified. Both of them show an E that's bigger than my original E. But it is only this one over here that has both been magnified, made bigger, and has high resolution, made clearer. Magnification and resolution. We're aiming for both of those every time we put a slide on our microscope. Not only do you want it bigger, but you also want it to be clearer. Magnification and resolution. When we're looking at slides, our slides contain 3D stuff. So here's an example slide that we have in our classroom. It's called the cross fibers slide. So think about literally 3 different colors of thread that we stacked on top of each other and put a cover sheet on. We have a 3D space that these 3 threads are inside of. When we're using our microscopes to make things bigger, our ability to see the 3D space changes based on which objective we have in place. So the amount of that slide that we're able to see clearly at one point in time is called the depth of field. Imagine that your slide is like a swimming pool. So how much of the swimming pool can we dive into? Can we see all of this clear? Or can we just see the little top part clearly? That's the depth of field. So when you have your scanning objective in place, the little red one, my depth of field is huge. I can see, in the case of this slide, I could see all 3 fibers. They're all clear. When I put that yellow objective, the low power objective in place, now my depth of field has gotten smaller. I can't see clearly through quite as much of that pool in my slide. Maybe I can just see 2 threads instead of all 3. By the time I put my high power objective in place, my depth of field is really, really small. So maybe I could only see part of one of my fibers. The depth of field, how much of that 3D space that I can get in focus, changes depending on which objective I have in place. So you may find sometimes when we're working with slides in the lab, you may find that you'll have to roll through that fine adjustment knob to play around in the depth of that slide. Maybe we can only see part of the top layer, but down a little bit lower is the tissue we're looking for. The depth of field, how deep in your slide you can see. In addition to how deep you can see, we also need to be aware of how wide we can see. So the field of view is how much space, wide space, on our slide we're able to see. We're looking at a picture here of a really boring slide. I am happy to report that in lab next week, we aren't ever going to look at something as boring as this black dot. But notice that when I start with the scanning objective in place, and then I go up to my low power objective, I've zoomed in on this spot. The spot got bigger. And when I go all the way up to the high power objective, the spot got really big. But here's what we didn't really notice happened when we zoomed in on that spot. We stopped seeing nearly as much gray on the outside of the spot. So the amount of gray that I see that's outside of my black spot goes down as I get more and more magnified. My field of view is essentially how much of that gray can you see? Well, the more magnified you are, the less of that outside stuff you can see. The reason that field of view matters is because you need to make sure as we go from the scanning objective to the low power to the high power, make sure that what you're looking at is in the very center of your field of view. If it's in the center, you'll still be able to see it. If we were setting up this microscope view and we decided to focus right here, if this was what it was in the middle of my field of view, when I zoomed in, that X might be over here. This would be where I zoom in. Whatever is in the middle of your field of view is what you zoom in on exactly. So make sure when we start using slides, don't leave it left or right, forward or backwards. Whatever you want to see needs to be in the very middle of your field of view. So you have a matching activity. We just talked about each of these terms, so we'll briefly run through here. We talked about the field of view. The field of view is how much of your slide you're able to see, how wide on that slide. We talked about the depth of field, which is how deep into that slide you're able to see, how much of the 3D we can see in focus. And then remember that our microscopes are parfocal microscopes. They mostly stay in focus when we go from one objective to another. That, them being parfocal, coupled with that small working distance, is why you can't use the coarse adjustment knob when the low power objective or the high power objective is in place. Again, I've talked way too much. I need to pause. What questions, thoughts, or emojis do we have for Dr. Allis? What would we like to ask? Let me get our party started here. Otherwise, we're going to have more crickets. I was so happy when I saw they had a microscope emoji. I like it, Jamel. That's a good one. Elenis, I love that you're doing the mind blown, because that's one of my favorite virtual learning emojis. Hopefully I didn't blow your mind too much, but it blown it in the best possible way, right? All right, I am pulling up something new for us. It's loading on my screen, and I'm giving you the link to this as well to help us see a little bit in 3D with these microscopes. I'm taking you to a website that has a virtual microscope. So I've put it in the chat if you want to follow along with me or if you want to play with it later. We're in a virtual lab working with a virtual microscope. So I'm going to explore here. We're going to play with the microscope a bit. When we look at this microscope, it's a lot like the pictures of the microscopes from our lab that we labeled. So you can see those oculars up at the top. You can see the objective lenses there on that revolving nose piece. We've got our stage where we're going to put our slides, and there's our stage clip to hold things in place. So we're going to play around with this microscope a little bit to watch the way that things work when we do things. So we're going to start here with, oops, unless it's dead. We're going to start with just a basic silly slide. We're going to start with the slide for the letter E. So I threw the letter E slide onto my microscope. This is what it looks like before I did any work to bring it into focus. Now notice right now, I have my 4 times objective in place. Remind me in the chat, the 4 times objective, what color is that one? What color was the one that made things 4 times bigger? Do we remember? Yeah, so the 4 times bigger one was the one that was called the scanning objective, and the scanning objective is the red one. So I've got my red objective in place. Since my red objective is in place, help me out. Yes or no on this coarse adjustment knob? Can I use the coarse adjustment knob since red is in place? I can. Yes, remember red is the short one. Red is the only one that I can use that coarse adjustment knob with. Now, I want you to watch something for me as I play with this knob. I want you all to watch my stage right here, the mechanical stage. So Dr. Allis is going to move around that coarse adjustment knob. Notice as I move that knob, how much the stage moves. It's going way up and way down. You will see this in our actual lab as well. If I roll this knob all the way around, it'll be in focus. If I keep rolling, it'll go out of focus. So when we're working with our microscopes, you want to roll your knob slowly till we get it mostly in focus. That looks pretty good. Now, with my 4 times my scanning objective in place, remember that I can also use the fine adjustment knob to get it just right. So I'm going to get it completely clear using my fine adjustment knob. I'm pretty happy with this letter E, so I'm going to go ahead and use my revolving nose piece to go up to yellow. Now I'm going up to the low power objective. Notice how my letter E stayed mostly in focus. That's because it's a parfocal microscope. All I need to do, remember, I cannot touch this guy. Coarse focus is done. Don't touch it. I can play around with this if I want to make it even a little more crisp. Because I have the yellow objective in place, I cannot touch this one anymore. Coarse adjustment knob off the table. Now that I've gotten it in focus with my low power objective, I'm going to go all the way up to my high power objective. Notice how little of that letter E I can see now because my field of view has gotten so much smaller. Remember, especially with blue down, I can only use the fine adjustment knob. And the other thing you might find in lab, I certainly always find this, I have to adjust the amount of light that's on my slide when I get up to that highest power objective. So I gave it a little more light. I'll play with that fine focus just a little bit more. Now notice if I go nuts either direction, I make it less clear. So I get it as focused as possible just using the fine focus or the fine adjustment knob. Cannot touch this one here. So we've looked at this funky letter E slide. Notice as I go down in my objective power, my field of view keeps getting bigger. I can see more and more of the letter E. Let's do one more slide briefly just to show you. We're going to go ahead and do a human slide. We're going to do a blood smear just because this one's easy for us to look at. I put this slide on my microscope, and this is going to be pretty typical. When you first put a slide on your microscope, what am I even looking at? There's nothing there. So we start with, since the scanning objective is in place, we start with the coarse adjustment knob. Notice I'm starting to see things. Okay, there's tissue here. I'm use that fine adjustment, get it as close as I can. That looks pretty clear to me. I can go anywhere on this slide. Notice how there's blood cells everywhere. But if I picked somewhere on my slide where there wasn't blood cells, when I zoom in, go up to the next power objective, I wouldn't have anything to focus on. So make sure what we're looking for is in the middle of our field of view. Now I'm going to go up to the low power objective. Now that I'm in the low power objective, don't touch the coarse adjustment knob, only use that fine adjustment knob. And with this particular slide, I'm thinking, yeah, I might want some more light. Okay, it's still very small. I need to go closer. I'm going to go all the way up to my high power objective. And now that I'm up on my high power objective, I'm going to give it some more light and use that fine focus to get it as crisp as possible. This is the same process we're going to go through where we played with each of these objectives. This is what we're going to do in lab together next week when we're looking at various different cells. We start on the scanning objective and work our way up from there. And I mentioned it before, I'm going to say it again. When we're in lab next week, if you've got your high power objective down and you can't for the life of you get things focused, go back to the low power objective. If you can't get it focused there, go all the way back to that scanning objective. Since we're parfocal, if we're messed up at the beginning, we're going to be messed up at the end as well. So we got to make sure to be aware of that. What do we think? Microscopes look easy, look hard. Can't wait to touch one, right? Oh, I got the crickets again. Y'all are killing me, man. Nice. Jamel gave me the easy button. Love it. Perfect. Yeah, Angelita, we will be in person next week. So we're gonna touch these microscopes. I think with practice, where it is gonna be easy for us. I think we'll be able to figure it out. All right. So we finished our lab class. Now we need to put our microscopes back. Let me help you talk through the way we'd put those microscopes back to do it properly. So the first thing we're going to do when we finish looking at slides in the lab, we are going to put our stage as low as possible. So we are not going to raise it as high as possible. We're going to drop it all the way down. That's going to help us have a really big working distance between our objectives and our stage. So we drop the stage all the way down. We take off our slides. Don't want to leave slides on the microscope. Put it back in our slide box. Then we're going to make sure that our scanning objective, the smallest one, is the one that's pointing down. We would never want that high power objective pointing down. That is the longest objective. We're going to bump into those slides, undoubtedly. So we put that scanning objective in place. With the slide off, with the stage dropped and that red objective pointing down, then we're going to go ahead and turn off the microscope and then unplug it. So we always want to power it off before we unplug it. There's a space in the arm, which is the part that sticks up on the microscope, that we tuck our cord inside of to store it so it's not moving all over the place. And then, like we mentioned for the beginning of the lab, we always want to make sure to use 2 hands to carry that microscope back into the lab cabinet. So 2 hands for transport. We will get so good at doing this at the end of every lab class. Drop the stage, put in that red objective, turn it off. It'll be easy breezy for us. So more practice with this to come starting next week. With that, I have talked with you about everything that I am related to microscopes. We pause and say, are there any last minute questions? Or give me a thumbs up if you're tracking with those microscopes. Angelita says this is great practice. Love it. Yeah, absolutely. Get us ready to use those things. I think I mentioned it. Microscopes are the most expensive thing we'll use all semester. So we got to take care of our babies, right? Perfect. All right, well, let's chat the last topic I'm gonna do with you, and then we'll go into group work. I got a last minute question from Jamel. Good question. What would I use the 100 times objective for? I believe you're asking, correct? For us playing around with the virtual microscope, yeah, so if you're playing with the virtual one, you'll see that it talks about a 100 times objective lens. That's called an oil immersion lens. For anybody in microbiology, that is going to be your domain. You're going to spend all your time with that one. When you're looking at things that are even smaller than human cells, which is like bacteria, for example, or yeast, you would want to use that even higher power objective lens for. If you use that one, it's called oil immersion on purpose. You have to put a little drop of oil on your slide. It uses that oil to make sure that the light gets into the really, really long objective accurately. So that we don't have 100 times on our microscopes in the anatomy lab, but you definitely do in the microbiology labs and some of our general biology labs as well. So it's out there. It's just not within the scope of our class. But great question. Yeah. I do love that virtual microscope. It's fun to play around with. All right. So let's talk cells. When we look at these 2 cell models that we have in our class, these 2 cell models should remind you a lot of that picture we looked at in lecture on Monday relating to the different organelles inside of our cells. So we're going to go through and label the big ones that we need to know on here. I'll mention some of the functions that they do. So let's go through kind of one by one and label things. We'll start here with the big organelle. And this is the one we started with in lecture as well. This big organelle that I'm putting a bracket around, this is the headquarters of your cell. This is where your DNA, which is your genetic information, is stored in. This big central organelle is called the nucleus. The nucleus. So your DNA lives inside the nucleus. DNA is the directions for every protein you will ever need to build during your cell's lifetime. So we got to store that in the nucleus to keep it safe. Inside the nucleus, you can see these little things, kind of looks like a marble over here, little white ball over here. These are what we call the nucleolus. Nucleolus. Think of it kind of like a nucleus inside a nucleus. So the nucleolus doesn't store DNA, but the nucleolus is what I use to help me build organelles called ribosomes. So ribosomes are my protein building organelles that I have in the cell. I actually build those little protein builders inside the nucleus using this organelle called the nucleolus. When we're talking about building ribosomes, let's introduce ourselves to those ribosomes. So all the little white dots that you see in this model on the outside of this organelle, and this conga line you see, oops, lost my pen, there we go. This conga line we see over here, this wavy pattern, these are all ribosomes. So ribosomes can exist in two places. Either they're free floating around in that soup of the cell called the cytosol, or they're attached, like we see over here, to my organelle that's called the rough endoplasmic reticulum. So this whole thing where we have these membrane flat sacs that are shown here with the green membrane and the blue inside, and then the little white dots on the outside. All of this is called the rough endoplasmic reticulum. So this is it too. It's called the rough endoplasmic reticulum because of those ribosomes on the outside. So I can see it over here as well. See all those places where we have the ribosomes on the outside, the rough endoplasmic reticulum. We also have in our cells, see these areas that are thin, that don't have the bumps on the outside. These are what we would call the smooth endoplasmic reticulum. The smooth endoplasmic reticulum. To be fully honest with you, I'm not convinced that the cell model on the left actually has smooth endoplasmic reticulum in it, because to me it looks like all of the endoplasmic reticulum has dots. But some people would say that this blue stuff right here is as close as we get to the smooth ER in this model. And I got another little section right here where there's none of those ribosomes on it. So as close as we get to the smooth ER in this model is right there. But to be frank with you, if I'm going to ask you about the smooth endoplasmic reticulum, I'm going to use this one over here. I really prefer this cell model for the smooth endoplasmic reticulum. Next, we have a couple of sets of organelles that are the same color on both of our cell models. So that's helpful for us. Let's start with our pink one that I see right here. The pink one that I see right here is the same as this pink one that I see right here. These are both representing the Golgi apparatus. The Golgi apparatus. And we talked about this a little bit in lecture, how this is kind of like the post office of a cell. So when I build proteins or I build other molecules, I send them to the Golgi apparatus. It packages them and sends them away. So the Golgi apparatus, they're in pink. We also see these orange organelles. They're orange on both of my models. Hey, these orange organelles are the powerhouse of the cell, as we said in high school. What is the name of these organelles that are orange? My powerhouse organelles, if you will. It's kind of mean for me to make you type that word, right? That's a long one. Absolutely right, though. These powerhouse organelles, the orange ones, these are the mitochondria. Remember from lecture, we said that their specific function was to make ATP, the energy that our cells use, ATP. One other set of organelles that I want to make sure to point out to you, and these are the fun food organelles, if you will. They look like foods to me. So right here, we've got our churros. The churros are the same as these little licorices that we see right here. These are things called the centrioles. Centrioles. So centrioles are the organelles that we use to divide up the genetic information of a cell. You will see these a lot. They're very important in what we talk about next week, which is called mitosis. So mitosis is the process where cells divide. We use the centrioles to make sure that we send genetic information into both of our new cells. There are some organelles that you'll notice in the word part. So you've got all these words here to match up with their functions. Some of the organelles that are listed here, you did not have to label. You just had to know their function, including a couple that I'm going to guess with you where they'd be found on the model, but I promise you I'm not going to actually ask you to identify them on a model. So the first one that I'm going to guess is these yellow ones that we see throughout the cell. I'm going to hypothesize that these yellow ones are those things we talked about in lecture called lysosomes. Lysosomes. Lysosomes were the organelles that help to digest things, to recycle things, get rid of waste. I'm guessing that that's what the yellow things are, but there's not anything for sure that makes the yellow things look different than the dark things. That's why I'm not going to ask you to identify them. I'm hypothesizing that these dark things are what we call peroxisomes. Peroxisomes. Remember that peroxisomes were my detoxifiers. These were the things that I told you were kind of like hazmat. So they come in, they clean up the mess, they protect your DNA from free radicals that want to come in and mess up with it. So here's a study, wink, wink, nudge, nudge for you. For any organelles that we don't ask you to identify on a picture, make sure you know what they do, because I'm limited in the number of questions I can ask you about those. I can only ask you what they do. I can't ask you where to find them. So make sure, especially for the ones that I can't ask you where to find them, because we don't have them on our models, make sure you know what those ones do. Are there any questions based on this content before we dive into our group work? I'm going to go ahead and stop the recording, and then we'll chat and we'll split up into our groups.